ce matin, nous accueillons Mathieu Cayla, qui nous vient de l'Université d'Édimbourg, d'Écosse. Aujourd'hui, il va nous parler des voies de signalisation qui régulent la différenciation sexuelle, la différenciation cellulaire, pardon, et la suivi chez les parasites kinoplastidés. Donc, Mathieu compte nous rejoindre d'ici quelques mois au sein de MIVEGEC. Nous espérons que cela va très bien se passer. Mais avant de passer la parole à Mathieu, nous allons écouter Cécile qui va nous parler des missions et des vols intérieurs. Cécile, nous t'écoutons. Et c'est parti pour la Minute Verte.
Bonjour à tous, merci beaucoup. Hop, je fais le partage. Voilà, donc aujourd'hui, le groupe MIVEGEC 1.5 présente une minute verte sur l'émission et en particulier les vols intérieurs. Donc, comme on le sait, l'émission représente la plus grosse partie, enfin une très très grosse partie du poids de notre bilan carbone. Et donc, quand ce sont des missions internationales, on n'a pas le choix, on va prendre l'avion, mais la question se pose lorsqu'on a des déplacements internes en France métropolitaine à faire, du choix entre le train et l'avion. Donc, tout d'abord, quelques recommandations. Quelques rappels sur les recommandations qui sont faites. La Convention citoyenne pour le climat qui a eu lieu l'année dernière recommandait un seuil de 4 heures. Pour un trajet en train inférieur à 4 heures, il est préférable de prendre le train. Lorsque le trajet est plus long, on peut alors prendre l'avion, mais peut-être en finançant des actions de compensation. Et les recommandations qui sont faites par nos instituts sont pour l'IRD de 4 heures également, mais pour le CNRS et l'université de 3 heures seulement, ce qui est moins contraignant. Vous vous rappelez qu'on a fait un sondage à la rentrée de septembre en interne à MIVEGEC pour avoir l'opinion des membres de MIVEGEC là-dessus. Donc, comme vous le voyez ci-dessous, une grande partie des gens étaient favorables à garder un seuil de 4 heures. Même certains étaient favorables à aller au-delà, donc à faire plus d'efforts et à fixer un seuil de 5 heures. Il y a aussi quelques personnes qui ont fait des remarques qui nous ont interpellés. Quand on calcule un temps de voyage, on ne devrait pas prendre en compte seulement le temps de trajet, mais le temps total qui peut être supérieur avec l'avion parce que souvent on ne prend pas en compte le temps d'acheminement qui est souvent plus grand que pour aller à une gare et le temps des démarches à réaliser qui est long également. Il y a aussi des personnes qui nous ont interpellés sur ce choix qui semble être recommandé de préférer l'électricité par rapport aux hydrocarbures et qui n'est pas forcément évident d'un point de vue environnemental. Ce qui est recommandé là, la base, c'est de diminuer l'impact carbone. Ce n'est pas l'impact global sur l'environnement qui est considéré, c'est bien l'impact carbone qui sera toujours inférieur en France avec l'électricité par rapport aux hydrocarbures. Je vais terminer je ne peux pas, c'est pas grave, je vais vous mettre dans le chat le lien vers un outil qui a été mis au point par Labo 1.5 qui permet d'évaluer l'impact d'une émission. C'est un petit calcul un petit outil qui permet de faire un calcul très rapide de sa mission. On peut le tester en faisant différents essais, donc en choisissant soit le train, soit l'avion, et de comparer les deux. Donc voilà, je vous mets le lien vers cet outil dans la discussion. Je vous remercie pour votre écoute et je vous souhaite un bon séminaire. Bonne journée.
Merci à toi, Cécile. Nous allons passer la parole actuellement à Mathieu Cayla.
Merci à tous et merci de me donner l'opportunité aujourd'hui de présenter le travail que j'ai fait pendant ma thèse et pendant mon postdoc à l'Université d'Édimbourg. Et je vais switcher en anglais maintenant parce que je suis plus à l'aise maintenant en présentant en anglais. Donc, I worked on all along my career on signaling pathway regulating cellular differentiation and survival in kinetoplastid parasites. And during my PhD at the Pasteur Institute, under the supervision of Gerald Späth, we focused in the understanding of signaling pathway regulating the promastigote to amastigote differentiation of Leishmania species, and particularly we focused on MAP kinases and tried to reveal the pathways they are regulating to allow adaptation and survival in the mammalian host. I was mainly involved in the study of one kinase, the MAP kinase 10, and I used different approaches to understand its function using a conditional knockout strategy and its regulation using transgenic and crystallization approaches. And today I will highlight some of the results we obtained with the transgenic approach, where we used an overexpression system based on the tagging of the MPK10 with the EGFP tag. So to study the activity of MPK10, I purified the tagged MPK10 from the parasites at different time points during the axenic differentiation. And I then revealed the kinase activity using a kinase assay where the MBP protein was used as a generic substrate, and I used a radiolabeled ATP. So after migration on a gel, I was able to reveal the phosphotransferase activity of the protein at different time points, as I said, by an autoradiogram. So on these plots here, you can observe the MBP loading control and the control of the MPK10 as revealed with the EGFP tag. And the result indicates that the kinase activity actually is sharply increasing during the first 48 hours of differentiation. And you can observe here on the right-hand side a quantification of these different plots that I performed. And this shows that in a logarithmic and stationary stage of the promastigote state, the activity is low, and it is increasing sharply during the first 48 hours of differentiation. And then this activity is reduced during the after 72 hours, which corresponds to the proliferation stage of the amastigote. And then it looks like it's increasing after 120 hours, which was corresponding when the parasites were reaching the stationary phase. So this result indicated us that the activity of the kinase was tightly regulated and during the differentiation, which led us to the question on what were the mechanisms involved in this regulation. So today I decided to highlight only one part of those results. And so where we used triptych digestion to reveal the kinase core. And the kinase core was nicely protected because it was nicely compact and it was poorly accessible for digestion. However, we identified a C-terminal domain that was easily digested. So we tried to understand what was the function of this C-terminal domain. And to do that, I generated parasites expressing either the non-mutated version of the kinase or the truncated version of this kinase that I called delta C. And the overexpression of this kinase had no effect in the promastigote stage. However, it was lethal during the differentiation in amastigote. And you can observe here on this graph the parasitemia that we measured during the differentiation. And on the right-hand side, you can observe the cell death that we measured as well. So as I said, with the overexpression of a non-mutated kinase, so a wild-type kinase, we observe a classical dip of parasitemia, which was accompanied by a high level of cell death in the population. And when the parasites that were able to differentiate were selected and started to replicate, as seen here on the gray curve, the cell death was going low. However, the delta C, the truncated version of the kinase, led to parasites unable to differentiate into stumpy form, into amastigote form, sorry. And it was followed by a very high level of cell death. And when I used a kinase dead version of this delta C by doing a mutation in the ATP binding pocket, by mutating a lysine into an aniline, which is a classical mutation to do to generate kinase dead, this restored the wild-type phenotype as observed with the triangle-shaped curve, where the parasites were able to replicate and the cell death was controlled. And so this suggested that the activity of the kinase was actually detrimental on later time points. And this was confirmed by this autoradiogram where the in promastigote, where the non-mutated form is barely active, and the delta C is actually very strongly active, which suggests that the C-terminal part plays an auto-inhibitory role. And this was confirmed by the kinase dead mutants that did not present any activity. In addition, we observed something quite surprising for us, is that MAP kinases contain an activation loop. And this activation loop contains two residues, usually a threonine and a tyrosine, that are targeted by upstream kinase to make this kinase active. And we observed that actually this motif, this TXY motif, was constitutively phosphorylated in promastigote, which suggested us that the kinase is actually constitutively active, but inhibited by a C-terminal domain. And what we observed, interestingly, was that the delta C version, which is non-controllable, a kinase that is cannot be auto-inhibited, was presenting a very low level of phosphorylation onto this activation loop. And this was linked to the activity of the kinase, because the kinase dead was not presenting any reduction of the phosphorylation. So this indicated us that there was a negative feedback loop linked to the activity of the kinase that tried to regulate the non-regulatable delta C kinase. So this result, as I said, indicated that MAP kinase-10 is pre-activated in a promastigote stage, and it is auto-inhibited with the C-terminal domain, which raised the question, what were the mechanisms controlling this auto-inhibition? So the C-terminal domain was poorly conserved among the orthologs of MPK10 in other kinetoplastids, except for a particular motif that I highlighted here that was nicely conserved. And this motif contained a conserved serine that actually we were able to identify as a stage specifically phosphorylating, and particularly phosphorylated during the promastigote stage, where it is dephosphorylated in the amastigote stage. Therefore, I performed the exact same set of experiments as previously by mutating this MPK10 on this serine, replacing the serine by an alanine to remove the phosphorylation site. As previously, no effect was observed in the promastigote, while in amastigote, I revealed a 24-hour delay in parasite growth and differentiation that is accompanied by a stronger cell death during the first 48 hours. We also revealed that this kinase activity, the kinase activity of this mutant was delayed, with a reduced activity observed at 48 hours during the amastigote differentiation, that then was 48 hours later at 96 hours was strongly activated. So this result indicated that the serine, the phosphorylation of the serine, is important for the right timing of the activation of the MAP kinase-10, deactivation of the MAP kinase-10, which allow a proper differentiation into a mastigote stage. So all the results that we have obtained, that I obtained during my thesis, allowed us to propose this model where in promastigote stage, the MPK10 is in a pre-active state with phosphorylation, a pre-phosphorylation of the tyrosine in the activation loop, and it is maintained auto-inhibited by the C-terminal domain, and the phosphorylation of the serine in the C-terminal domain seems to be playing an important role for the regulation of this auto-inhibition. And upon the extracellular signal, like the pH, lowering the pH and increasing the temperature, the parasites are initiating the differentiation, and an upstream kinase, a MAP kinase, would phosphorylate the threonine on the activation loop, and the phosphatase will dephosphorylate the serine on the C-terminal domain, this relieving the auto-inhibition of the kinase and rendering the kinase fully activated during the first 48 hours. Then the kinase needs to be deactivated, otherwise it's lethal for the parasites. And so works remains to be done here to identify the exact mechanism, but we were able to see that there was a dephosphorylation of the tyrosine, and whether or not there will be the auto-inhibition again by the C-terminal domain remains to be identified. So then I joined Mathieu's lab in Edinburgh to study signaling and phosphorylation during the quorum-sensing dependent differentiation in the parasite Trypanosoma brucei. And so in the mammalian bloodstream, as a slender form proliferates, they are initiating a differentiation process in a quorum-sensing like dependent manner in response to an accumulation of a Stumpy induction factor or SIF. And recently, this Stumpy induction factor has been identified. And it resulted from the paracrine secretion of peptidase from the parasites that were able to digest host proteins that generated oligopeptides. And these oligopeptides were sensed by the parasites. And when a certain amount of oligopeptides were sensed, this initiated a differentiation process in a quorum-sensing manner. And this differentiation is characterized by the exit of the proliferative cell cycle with a G1, G0RS, and a differentiation into a short stumpy form, expressing the PAD1 marker on the cell surface. And these parasites are adapted for transmission by the insect tsetse fly. And the molecular composition of the pathway that transduce this SIF signal to induce the stumpy differentiation starts to be quite well characterized. And this is because the action of this SIF signal can be mimicked in vitro by cell-permeable cyclic AMP analogs, which causes monomorphic or cell lines to arrest but not undergo a full development stumpy form. And this enabled previous postdoc in the lab to perform a genome-wide RNA-I selection for a molecule whose silencing renders the parasite unresponsive to the SIF signal. And they were able to identify several components of this pathway, which contained actually several protein kinases. And I was particularly interested by this kinase here, that blast analysis revealed to be related to YAC family, where a number of proteins have also been identified in differentiation in yeast and Dictyostelium, for example. So they perform a knockdown of this gene coding for this YAC kinase, and the knockdown of this gene actually renders the parasites unresponsive to the accumulation of the things, generating an uncontrollable parasitemia here in red compared to the white type cells that were arresting and they were controlled by the mice. Then I performed a phylogenetic analysis using the kinase score of the CMGC kinase family members of different species. And I revealed that this kinase indeed belongs to the DER group, but it's branching quite differently to the other subgroups that have been identified, suggesting high divergence. But we can note that actually, it's not visible here, but we can note that it's branching with two Cenorhabditis elegans kinase that have been annotated as part of the DIAC2 subfamily. And then when we look in more detail, we observe that this kinase present a large N-terminal domain where no homology domains were identified except for a divergent NAPA1 domain and a divergent DH box that both have been characterized in DIRC2 subfamily and have been shown to be implicated in the generation of an intermediate form of the kinase that is able to autophosphorylate during its own translation. And then the two phosphorylate on actually on a tyrosine, and then the specificity of the kinase change when it's fully active and fully translated, and the kinase becomes able to phosphorylate serine and threonine only. This N-terminal domain also contains several phosphosites that we and others have identified, and I highlighted them with the stars here. And the C-terminal domain actually contains a classical kinase core. However, this kinase core is interrupted by 3 big inserts that are uncharacterized. In addition, this kinase possesses a very interesting and unusual amino acid into highly conserved and essential motif amongst kinases usually. Particularly, we can identify a serine in this DFS motif that is usually a very conserved glycine that form a DFG motif, and I will come back on that later. We also identified HXY motif in the activation loop, and as I said, usually activation loop are formed by 2 phosphorylable residue, and particularly in this kinase family, it's 2 tyrosine. And as I said, the second tyrosine is the one autophosphorylated, and the first tyrosine is the one targeted by the upstream kinase to render the kinase fully activated. So one of the main goals of my project was to perform structure function analysis of all the sites that have been highlighted here in red, and to understand their role in the kinase activity and function. So to do so, we first observe the growth phenotype of cell ectopically expressing mutant form of this DIRC kinase, and we observe the phenotype in response to the Stumpy-like inducer cell-permeable cyclic AMP analog. So we can observe that in Y-type cell, the incubation with the cyclic AMP here in red drives the cell into Stumpy-like differentiation with a cell growth decrease. In the knockout cell line for the gene coding for this TB-DIRC, this knockout cell line showed a resistance to the cyclic AMP analog, and when parasites were ectopically expressing a non-mutated kinase version of the kinase, this renders the parasites actually more sensitive to the cyclic AMP analog in vitro. And this phenotype was fully reversed when performing a mutation into the ATP binding pocket to create kinase dead. And this actually ectopic expression of this kinase dead led to a dominant negative phenotype that is similar to what observed with the knockout cell line. So then we needed to know what was the activity of each mutant of this kinase. So to do that, we purified the recombinant DIRC kinase carrying the different mutations from insect cell and tested the activity by in vitro kinase assay against this time the Caspase-9 as a generic substrate, and we revealed here the mutation. And this revealed here that the mutation containing the lysine, the mutation of the lysine 592 into an alanine into the ADP binding pocket leads to a loss of 50% of the kinase activity in vitro. So with this set of experiments, we investigated the effect of several mutations for the activity of the kinase. And as for example, I will present the result of the unusual DFS motif of this kinase. So as I said, this motif usually contains highly conserved glycine and this is highly conserved among the other species and other kinases. And this glycine actually plays a very important role in the mobility of this motif, because upon activation by phosphorylation by an upstream kinase, this glycine allows the movement of a phenylalanine here from the DFG out position, and that move to a DFG in position here shown in gray. And this aligns and allows to the kinase to bind ATP because it forms a catalytic spine. When I modelized the kinase core of the TB-DIRC, I observed that actually the serine will rigidify the kinase and force a pre-active state in a DFS in conformation here shown in red. So then I tested this hypothesis using the same pipeline as described earlier, and I investigated the effect of mutating this serine either into an aniline to remove the phosphorylation site, or by mimicking the classical glycine. And both of those mutations restored the wild-type phenotype as observed on this growth curve in response to the cell-permeable cyclic AMP analogs, which suggested that both of these mutations render the kinase inactive. And this was confirmed by the kinase assay for the serine to glycine mutation, which completely abrogated the kinase activity. However, with the serine to alanine mutation, the kinase keeps around 70% of its activity on the caspase-9 substrates. And this is interesting because the alanine is a less mobile amino acid than the glycine itself. And so it allows intermediate conformation, but not as much as the glycine. And this would suggest that actually the conventional flexibility brought by the glycine is actually detrimental for the kinase activity of TB-DIRC, and the serine will stabilize the kinase activity in a pre-active conformation state. So on this slide, I'm summarizing all the mutations that we tested using the same methodology, and basically all the residue that we have mutated were essential for the kinase activity. Even the phosphorylation sites that were far away in the N-terminal domain that were potentially also implicated in the regulation of the kinase activity. Both residue, even the histidine, which is a non-phosphorylable residue on the activation loop, were essential for the kinase activity. The three inserts that are actually disrupting the kinase core were also essential for the kinase activity, and they were potentially implicated in protein-protein interaction, as the structures seems to reveal that they were not in the core, but they were maintained outside of the kinase core. And finally, the DFS motif, here in purple, was leading to the pre-active state of the conformation of the kinase. So the next step of this project was to unravel the potential substrate and the pathway in which this TB-DIRC was involved. So to do that, we used two different and complementary approaches. First, we used knockout cell line for this kinase and compare its phosphoproteome to a wild-type cell line. And to do so, each replicates were labeled by a 6-plex GMT isobaric tag, and the phosphopeptide were enriched and are analyzed by a quantitative mass spectrometry. In parallel to that, to filter a bit the list of proteins that we obtained, we purified from insect cell an active kinase as well as an inactive kinase, and we performed direct in vitro kinase assay on parasite lysates as substrates. And the resulting phosphoproteins were then differentially analyzed by mass spectrometry, which give us these two volcano plots. Or on the left-hand side, you can observe the phosphoproteins that are changing in the knockout cell line compared to the wild-type cell line. And on the right side plot, you can observe the phosphopeptides that were generated by the active kinase compared to the inactive kinase. And we then compared the list, identified common phosphosites to determine the substrates of this TBDIRC. So then we selected some of these substrates and we tried to see if they were implicated in the slender to stumpy differentiation. And to do that, we generated knockout or knockdown cell line. And as an example, on one hand, you have the deletion of the zinc finger protein, ZC3H20, led to parasites that were not during in vivo infection, that were not responsive to the stumpy differentiation factor, and they were not expressing PAD1, not differentiating at all, and they were maintained in a proliferative stage with no accumulation into G1, G0 state as represented by the kain counting. So this suggested that this ZC3H20 is actually a stumpy inducer protein. And on the other hand, the knockdown of this gene coding for an hypothetical protein led to parasites that were actually more sensitive to the cyclic AMP analog. So this suggests that the knockdown of this gene pushes parasites to become stumpy at a lower density, which indicated that actually this gene is coding for a slender returner protein. So the next step was to confirm that there was a direct link between the TBDIRC and the substrates that we have identified, and if the phosphorylation by the TBDIRC was influencing the stumpy differentiation. So to do that, I endogenously mutated parasites on the ZC3H20 to remove the phosphorylation site. And as we determined that the ZC3H20 is a stumpy inducer protein, we expected to observe that the proteins that were not able to phosphorylate will not able to differentiate into stumpy. And this is exactly what we were able to observe with the parasite expressing the non-phosphorylable version of the ZC3H20 that were becoming hypervirulent in mice, not expressing the PAD1 differentiation marker, and they were maintained in a proliferative stage. So all these results led us to propose this model, this working model, where upon reset of the CIF signal, the TBDIRC will become activated and will phosphorylate stumpy inducer proteins and activate this protein like the ZC3H20, but will act as well on the other hand of the differentiation pathway by phosphorylating and inactivating stumpy slender returner protein. And this led us to the hypothesis that the TBDIRC will actually work as well through the regulation of stress granules that may regulate developmental gene regulation. So why did we hypothesize that? It's because the ZC3H20 protein first have been shown to be a gene expression regulator. And in addition, our phosphoproteomics analysis presented an enrichment for protein implicated in post-transcriptional gene regulation. And actually, several of the components have been identified into stress granules. So how does the TB-DIRC link to gene regulation? Several papers has also, in fact, several papers have implicated kinases, and more particularly DIRC kinases, in stress granule dynamic regulation. So before I go in more details, so what are stress granules? Stress granules are membraneless organelles that are composed of RNA that are maintained in a silent stage and proteins, and they have been hypothesized to protect mRNA while the organism were dealing with the stress. And these stress granules are formed by liquid-liquid phase separation through the presence of low complexity regions called LCR on the proteins. And TBOCI have been shown to present stress granules upon nutrient starvation, for example, where PABP2 and ALBA3 has been published as forming stress granule, as you can observe here on this immunofluorescence, as well as other proteins. So during the lockdown imposed by the current outbreak, I initiated a bioinformatics project because I was stranded at home, where I proposed to identify low complexity regions on the proteomes of Trypanosoma brucei to help me to filter from the list of potential phosphosites of TB-DIRC for proteins that contains low complexity regions, so potentially involved into stress granules. So what we observed was first that the proteome of Trypanosoma brucei contains 42% of the proteins in Trypanosoma brucei contains low complexity regions. And actually, this makes Trypanosoma brucei one of the organisms with the highest percentage of low complexity regions, with dictyostelium that contains around 50%, while the classical other models like yeast and humans contain less than 10% of low complexity regions in their proteome. And what we observed as well is that there was an enrichment of low complexity regions in the extremities of the proteins. And interestingly, when we looked at the function of the proteins that were presenting low complexity regions either in the N-terminal or the C-terminal part. So in the N-terminal part, we revealed that the protein were enriched for enzymatic activities like cyclase, kinase activity, transferase, while the proteins that were presenting low complexity regions in their C-terminal part were actually enriched for nucleotide binding function. So the localization of the low complexity region seems as well to be a way to select for protein activity, different type of activities. And as I said, protein kinase have been implicated into the regulation of stress granules because phosphorylation is changing the interaction between low complexity regions and allow aggregation or actually dissolution of stress granules, as well as other post-translational modification. So we looked into the datasets that have been published for all the post-translational modification in Trypanosoma brucei, and we identified actually that the phosphorylation were the post-translational modification that was highly enriched into the low complexity regions compared to the rest of the proteome. So this leads us to the hypothesis that actually phosphorylation will be a way to regulate low complexity regions by phosphorylation. So we used these data sets that we generated, and these two proteins that have been previously shown as to be implicated into stress granules to reveal. And this, you can observe here on these graphs that PAB2 possesses 3 different low complexity regions, and alba-3, one big on its C-terminal part. And the idea was to tag this, endogenously tag this protein and to look for the formation of stress granules during the stumpy differentiation in vivo. So this is what I did. And on this immunofluorescence, I present a time course of the, where I followed the parasite during the differentiation to stumpy stage into the mice. And I revealed the differentiation through using the PAD1, looked for the localization of alba-3, and I used DAPI as a marker for DNA. And on this panel, you can observe that while the cells are slender, so early into the infection, they are presenting a weak expression of alba-3 that is diffused into the cytoplasm. However, when they are starting to express PAD1, so they are starting to initiate differentiation, parasites are expressing granules containing alba-3. And this was shown as well at day 6. And if we looked in more detail into the stumpy differentiation, so we are using different markers. For example, as well, one marker is the distance between the kinetoplast and the nucleus. And early stage of stumpy are presenting a long distance between this kinetoplast and nucleus that is reducing into intermediate stage, and into late stumpy stage, they are very close together. And interestingly, what we observed was that in early stage, the granules were located posterior to the nucleus, between the kinetoplast and the nucleus. And while they are progressing into the differentiation stage, so in the intermediate, while the nucleus was getting closer to the kinetoplast, the lower granules were located then around the nucleus to finally end up at the anterior end of the nucleus into the late differentiation stage. So then I did the same exact experiment, but this time, instead of using a white type background, I used TBDR knockout background, and looked for the presence of PAD1 and the distribution of alba-3. So as you can see, the cells are not differentiating into stumpy because they are not expressing PAD1. And interestingly, I was able to observe that the localization of alba-3 was completely disturbed. For example, I could see some localization around the nucleus, still at a weak level, or later time point diffuse into the cytoplasm, and then it was disappearing at later time points, which suggests that TBDR may be involved in the proper localization and expression of alba-3 as well. As a control, I also used this PABP2 that I said have been shown to be implicated into nutrient starvation stress granules, glucose starvation stress granules, sorry. And I performed exactly the same experiment that has been done before by tagging a PABP2 that is in rich glucose condition, diffused and highly expressed into the cytoplasm. While in minus glucose condition in starvation, they are expressed, PABP2 is localized into stress granules that is diffuse, that are all around the cytoplasm. But interestingly, when I performed the mice infection, I observed that the PABP2 was actually not going into stress granules, but was maintained diffused into the cytoplasm, which suggests that the stress granules from the glucose starvation and the stress granules formed during the stumpy differentiation are of a different nature. So this is where I am right now, and this is still a work in progress, where alba-3 and alba-4 are diffused into the cytoplasm into slender form, the same as PABP2. And when these cells are stressed by starvation, they are forming stress granules that contains these 3 proteins that are all around the cytoplasm. However, while the cells were differentiating into stumpy, the PABP2 protein was maintained into the cytoplasm and diffused, while ALBA3 and ALBA4 were relocated into stress granules that were located between the cytoplasm, the nucleus, and the kinetoplast in early stumpy stage, and that was moving anterior to the nucleus in later stages. So I'm trying to still understand what is the exact function of TB-DIRC in this role. And for that, I am trying as well to identify what are the components, because they seem to be different from the one from nutrient starvation granules. What are the components of these granules in TB-DIRC during the T-brucei during the differentiation process? So I'm done with the presentation on what I have done so far. And I wanted to take the opportunity today to as well present the work that I would like to develop in the future. And this will look at the signaling and regulation of autophagy-related pathway during the differentiation of African trypanosomes. And I presented these slides earlier, but here I just added that these parasites contain 190 kinases, and several of them have been implicated into the differentiation process. So what is autophagy and the related pathway? So the autophagy is a process that is essential for maintaining cellular homeostasis, where protein aggregates, parasites are targeted to the lysosome for degradation. And it has been shown that this autophagy-related pathway are involved in cellular differentiation. And very interestingly, there is an alternative secretion pathway that is linked to the autophagy, because this is called lysosomal exocytosis. And this exocytosis allows the secretion of the content of the lysosome into the extracellular medium. And this pathway contains marker of autophagosome and actually is sharing machinery with the autophagy mechanism. And this pathway has been shown, for example, to release peptidase directly into the extracellular medium that allows the digestion of cell wall to allow Caenorhabditis elegans to invade tissue. So it has been implicated in tissue invasion. And autophagy and related pathway have been shown to be regulated by kinases. And interestingly, Trypanosoma brucei is lacking some of these kinases that have been implicated in the regulation of autophagy and or are independent of the classical ones that have been identified. So I'm asking the question, what are the signaling pathway regulating autophagy-related pathway in Trypanosoma brucei? And then I want to understand what the function, what function do the autophagy-related pathways play during Trypanosoma brucei differentiation and for the tissue invasion. So first of all, to develop this project, I needed preliminary data. So, and I was able to observe that autophagy is actually essential and tightly regulating during the stumpy differentiation. And to observe that, I use wild-type cell line, where I endogenously tagged this ATG8.2 protein. And this ATG8.2 is an ortholog of the ATG8 that is a hallmark to observe autophagosome formation. Sorry. Oh. Sorry, yeah, there's a machine going on. Sorry for that. And so, using this M-cherry tag protein, I was able to observe that the autophagy is initiated at the intermediate stage, only for a very short period, where in stumpy form it is disappearing. So I have quantified this autophagosome formation using particle countings. And you can observe on this graph that there is an intermediate activation of autophagosome formation during the differentiation of the parasites, just when the parasites start to express the PAD1 marker cell line. And then it is reduced at the next day of infection, where the parasites are almost fully stumpy differentiated. And interestingly, when I tried to disrupt this autophagy pathway to see if the autophagy was implicated into the differentiation pathway, by deleting this ATG5, I was not able to obtain a double knockout for this one, but the single knockout was actually enough to disrupt completely the autophagosome formation during the infection. And as well, I was able to show that this disruption of the autophagy pathway was impairing the stumpy differentiation. As you can see here, only 25% of the population is expressing the PAD1 marker, and they have been eliminated by the mice on later time points. In addition to that, I wanted to see if I was able to observe lysosomal exocytosis during the differentiation during the stumpy differentiation. And to do that, I used a stumpy differentiation system that we developed in the lab, an in vitro one. And I did, I tagged two different proteins, synaptotagmin 7, which has been implicated into the fusion of lysosome membrane with the plasma membrane, and the VAMP7 as a marker for endosome and lysosome. And I did perform two different conditions after exposing to the in vitro differentiation system, where I permeabilize the cell to see the localization of this protein in the cell, or did not permeabilize the cell to see if there was a presence of this protein on cell surface. And I was able to observe that the synaptotagmin is slowly increasing during the differentiation process inside the cell, while I was able to observe on the cell surface an intermediate staining of the synaptotagmin 7, which will suggest fusion of lysosome and exposure of this synaptotagmin 7 on the cell surface of the parasite, which may be an indication of lysosomal exocytosis. And similarly, using the lysosome marker, I was able to observe lysosome formation during the, actually lysosome during the differentiation process. And interestingly, the same, I was able to observe an intermediate localization of the VAMP7 on the cell surface of the parasites that is decreasing on later time points, which will suggest potential lysosomal exocytosis during the differentiation pathway. So in this project, I would like to develop a three-pronged approach, where first I will look at the autophagy-related kinome and identify the kinase implicated in that autophagy processes. And I will develop a high-throughput live imaging system that will be combined with a kinome-wide RNA screen, and that will be based on the expression of this dual tag ATG 8.2 using mTOR and EGFP that will allow me to distinguish autophagosome and autolysosome, because the GFP signal will decrease when the autophagosome will reach the lysosome, because the pH will quench the fluorescence of the GFP. The second approach, we looked at the dynamic autophagy and lysosomal exocytosis proteome and phosphoproteome, and this will be based on a APEX2 proximity labeling system that will allow me to identify the component of the autophagy in Trypanosoma brucei. And I will use that to as well purify phosphoproteins and using knockout or wild type cell line or mutant to identify the regulation by phosphorylation of these pathways. And then we'll perform kinase assay to understand, to reveal the direct interaction by the kinase identified in the objective 1 with the substrate identified in the objective 2. And then the third objective, we look at the autophagy and lysosomal exocytosis regulation during the differentiation for the tissue invasion. And for that, I will use a different system using in vitro differentiation system, or using mouse for the mammalian model, or the tsetse fly infection for the model of the insect vector. And I will perform functional analysis of knockouts and mutants. And this together will reveal the interplay of autophagy and exocytosis for the differentiation and tissue invasion. And as a proof of principle for the proximity labeling, I performed an experiment where I exposed cell to amino acid starvation to initiate autophagy at different time points, and I pulled down the biotin-related protein, proximity protein of the ATG 8.2. And then on this plot, you can see that there is change in the biotin-related protein during the autophagy induction, where protein are disappearing at from 30 minutes to 60 minutes, and other protein appearing at later time points. So that seems to be quite a dynamic proteomes to be revealed here. So altogether, this project will uncover novel biology of trypanosome parasites by revealing how protein kinase regulate autophagy on lysosomal exocytosis to promote parasite survival and host adaptation with quorum-sensing dependent differentiation and tissue invasion, and what will be the role of the peptidase and really what will be the role of lysosomal exocytosis for the peptidase and release of hydrolase, inducing the stampede differentiation, and potentially the role for the receptor recycling, like the GPR89. And that's it. I would like to thank you for your attention. I would like to thank as well the labs in which I have performed my work. So Gerald Späth and Najma Rashidi and other people implicated in this project. And Keith Matthew's lab, with Lindsay particularly as well involved in this project, and Paula McGregor, Al Evans, and Philippe Bastin. Thank you.
Merci à toi, Mathieu. Bon, actuellement, nous allons accueillir des suggestions, des questions dans le chat. C'est free.
Mathieu, je peux poser une question directement?
Oui.
So a question about your low complexity regions, which I found very interesting. Presumably you did all that on a single strain of trypanosoma.
Yes, we looked only on the 927, but we generated a script that is actually we can use for other organisms actually.
Okay, that's interesting. Because for these low complexity region, they tend to accumulate in insertion and deletion quite regularly.
Yeah.
So there will be the length of the LCR will be quite different from one isolate to the next. So do you think that could have, so different isolates could have different level of phosphorylation?
That is a good question, and I would imagine so that actually like virulence could be probably affected by the level of phosphorylation, because if some cells are more or less responsive to differentiation, they are more or less virulent. So I would imagine so. But from the data I have, mainly the phosphorylation that have been done were done in the polycyclic stage and or slender stage, and on one species. So it will be complicated for me to give a straight answer saying yes or no. But I will imagine so that absolutely it may play an important role. It will be interesting to see if there is differences between low complexity regions between different isolates. Absolutely.
How many genomes are available for trypanosoma? I have no idea.
For the reference genome, there are probably 10 reference genome, but there have been quite a lot of isolates for these genomes that have been used. And actually, yeah, it's a very good question. It could be something that could be done. Absolutely.
Thank you.
Thank you.
Okay. Merci, Antoine. Une autre question ou préoccupation?
Je peux répondre en français aussi, il n'y a pas de problème.
Vous pouvez aussi laisser vos questions dans le chat.
Maybe just to continue on the LCRs, you also have to pay attention on the quality of the assembly of the different genomes, I think. Yes. This is very important, and I don't know, so I think this is a very important point. In particular, if you compare genomes from different origins, quality of sequencing has to be the same, because if not, you could infer things.
Yeah. Yeah, the annotation need to be right, because often if the annotation are missing the some N-terminal or C-terminal part because of mislabeling of the start codon, actually you will miss the important information about the extremity of the low complexity regions.
I don't know how many PacBio or new assembly genomes are available from, for example, from Nicolas Siegel. Like that.
And I'm not sure Nicolas Siegel is too much involved in.
No, I'm sure he's not involved in the LCR, but he's really involved in the new assembly.
Yeah, yeah. So there are like very good assembly for the 427 now that he has made. But yeah, it will be good to have a look for as well the field strains, as Antoine suggested.
So I actually have one question as well. Bonjour Mathieu, merci pour ta présentation. Just a few questions regarding the project that you want to do regarding autophagy. Yes. You were quite fast, so I didn't really get on which protein would you fuse the apex?
So sorry, continue.
The apex, you're going to fuse it on which protein?
So as a start, I wanted to start with the ATG 8.2 because it's a reference protein, and actually this is what I showed on the blot. Okay. I started with this one using ATG 8.2, but there are as well other proteins that have been identified as part of the autophagy pathway, for example, ATG6 or ATG15 that plays at different time points during the autophagosome formation, and it will be really interesting to look at these different time points, these different proteins to reveal different time points and different composition according to the stage of development of the autophagy.
Okay, but the blot that you showed, it was the bioattenuation profile when the apex was fused to ATG8.
Yes, absolutely. Sorry. Yeah.
So, because you showed that you have, well, you said that you have different profiles, but would it means that you have different profiles because the ATG8 is differentially expressed when you have starvation induction, because you showed the localization of ATG8 and you sometimes show that ATG8 is not expressed at some different point of this intermediate or STUMP. I don't remember. So the different profile that you got is probably due to the fact that sometimes ATG8 is not expressed at a certain stage of the.
So there are like so the ATG8 looks to be constantly expressed. I did not show that, but there are like a publication where you can find the expression of different proteins. It looks to be constantly expressed. However, what I'm seeing is when it's condensing. And when it's condensing, this is where I'm able to reveal it because it's not necessarily highly expressed. But when it's condensing, this is where I'm able to see it by immunofluorescence. So it's more like the different profile that I see is probably the other proteins that are interacting around it that are changing while the ATG is getting localized into autophagosomes or autolysosomes on later time points. And yeah, sorry. Yeah.
No, go ahead if you want to just, because I'm going to ask all the questions.
I was just about to say that ATG8 is actually localized on the autophagosome, first outside and inside the autophagosome that are forming. Then it is cleaved out from the outside, and what we will reveal is probably the proteins that are still inside the autophagosome because the ATG8 is still there on later time points of the autophagy.
Okay. And just regarding the tagging that you wanted to do with ATG8, the double tagging with mCherry and GFP, so have you already tried to use it and just try to see what's going on with the labeling with GFP when you induce starvation? Does it work?
No, unfortunately, I haven't done it. Somehow I'm struggling to get the eGFP into the plasmid. I don't understand. It drives me mad. It should be very simple, but it does not go into the plasmid. But I think I got it like 2 days ago. So now I will be able to try to see in the parasites if I'm able to see the difference. But it has been published in other organisms, this system.
Yeah.
So in theory, it should work.
Okay, so just was actually quite interesting about your result, the first result that you've presented regarding the MPK10 in Leishmania.
Yeah.
So you show actually different activity in amastigote and promastigotes, and I was wondering if you were able actually to know whether this kinase was involved in the differentiation of promastigote into amastigote per se, or if it was involved in the development or division of the amastigotes.
So my answer to that will be, I don't have a defined answer because we have struggled to identify the substrate for it. But my answer to that will be, it is activated during a short period during the first 48 hours, while the parasites are dying or adapting to that. So to the stress. So my answer will be that it's actually for the differentiation itself. And it needs to be, the activity needs to be reduced for the parasite to proliferate, because the delta C, which cannot be inactivated, leads to the parasites that are dying. So yeah, my answer will be, it is during the differentiation and not for the proliferation of amastigote.
Okay. So what will you expect if you would delete the gene?
So it's essential. So that's why I did the conditional knockout approach. And so what I was able to see was actually, so for the conditional knockout approach, I expressed the kinase on the topically on the plasmid, and then I deleted the 2 alleles. And then I'm removing the drug that I maintained for the maintenance of the plasmid to see if the parasites were slowly able to lose the MAP kinase 10. And actually, I was just able to see, I saw that they needed to maintain the MPK10 constantly, and it was essential for differentiation. I could not remove it.
Okay.
Maybe with CRISPR, because at the time I did it, there was no CRISPR. So with CRISPR, it's a faster knockout instead of doing like a long run of selection with maybe it will work better. And you could have a window where you could observe stuff, but I'm not sure.
We also developed an inducible CRISPR.
Yeah, I've seen it. Yeah. At the time, there was no CRISPR.
No, absolutely, but you said that you didn't see a decrease on your episomes, but did you have a control? Because in our experience, decreasing of episomes are very low in Leishmania.
So I had a control with like where I put the same plasmid, but without the gene containing a EGFP tag, and I was following both. So I could see that the plasmid containing only the EGFP was decreasing really quickly during the replication. So they were losing it because it's not essential. While the kinase, they were losing roughly half of the amount, but it was an overexpression system. So until it stabilized basically to half of the amount that was initially maintained with the drug. So, and then I could not go lower.
Okay, so this is interesting, and we may contact you or Gerald, because I think Gerald still has the plasmid.
Yes, definitely. Yeah.
And Okay, this is nice.
PXNG system that Stephen Beverley published at the time.
Okay, okay.
Okay, and so would you expect that this function is conserved in trypanosome, like you were showing an alignment with the sequence that is quite conserved? So would you expect that the function would be conserved in, for example, trypanosome for the differentiation into from slender to stumpy, or?
That is a very good question. It is conserved, it is there in trypanosome. What I've seen is that actually the temperature was a trigger for rather than the pH. So I may imagine that it has a similar function because trypanosome will be exposed to this change of temperature while it will not be exposed to the change of pH because it's not internal. The parasite is still in the blood, in the trypanosome. So I would imagine that it has a similar function because the protein was actually the trigger for the activity and for the expression was the change of the temperature when they were put into the amastigote stage. So yeah, I would imagine that there is a similar function, but yeah, no one, it's like all the kinases that has been published, no one looked on both organisms, even if they are conserved. So you need to infer potential function from one to the other, but sometimes it's complicated.
Okay, thank you, Mathieu.
Thank you.
I guess we will have opportunity to talk a bit more after. Thank you.
Thank you.
Merci, Mathieu.
Merci beaucoup.
Merci à tous les intervenants et merci à ceux qui ont suivi le séminaire. La semaine prochaine, jeudi le 16, nous accueillons comme chercheur Cayla Deschavannes qui nous vient du CRCN. Et ce séminaire sera tenu par nos 3 nouveaux membres de l'équipe d'organisation des séminaires qui vont se présenter tout à l'heure. Ils vont prendre le relais et vont assurer l'organisation des séminaires tout au long de l'année 2022. Il s'agit de Sophie. Je crois qu'elle est là. Vous pouvez activer vos caméras si vous le voulez bien.
Bonjour à tous.
Sophie, tu peux te présenter en 1 minute et faire un coucou.
Donc moi, je suis Sophie Tissot. Je viens de débuter ma thèse donc en septembre. Je fais partie de l'équipe de Frédéric Thomas qui travaille sur l'écologie et l'évolution du cancer et notamment donc sur le modèle biologique Cayla. Voilà.
Ok, merci Sophie. La suivante, Margot le fait. Margot.
Bonjour à tous. Vous m'entendez?
Oui.
Merci. Bonjour à tous. Moi, je viens aussi de commencer ma thèse en octobre. Je suis dans l'équipe avec Florence, avec Virginie Rougeron et Franck Rignol, qui sont partis en Afrique du Sud, comme vous savez. Et je suis aussi avec Michael Fontaine. Et je fais surtout de la génétique des populations sur des plasmodiums.
Et en 3e position, nous avons Thibaut.
Oui, bonjour. Du coup, moi, je travaille avec Karen McCoy dans la même équipe que celle de Margot. Donc, ensuite, elle va nous quitter, mais moi, je vais rester dans cette équipe-là. Et donc, moi, je travaille sur la dynamique de transmission des pathogènes chez les goélands. Donc, voilà.
OK. Donc, ce sont ces 3 nouveaux visages qu'on verra à partir de jeudi prochain. Tout de même, nous aimerions bien vous annoncer que nous avons besoin des volontaires pour la séance du 6 janvier. C'est vrai, c'est la première semaine, mais bon. Entre-temps, tout volontaire serait la bienvenue. Donc, n'hésitez pas. Donc, on vous dit merci. Merci à tout un chacun et bon appétit à tous. Une fois de plus, merci à toi, Mathieu.
Merci.
Merci. Bon après-midi.